Hello, my name is Wang, and today I'll be talking about fungicides. When you take a stroll in your garden and see plants that look like this, if you're a garden enthusiast, you know something is wrong. In fact, the plants are sick. And in fact, the plants shown in the pictures are infected by the parasitic plant fungi, which are very common pathogens for plants. Many of these parasitic plant fungi affect the plant's leaves, and which makes it immediately visible, and they come in a variety of forms. Some of them will blush the plant's leaves, as the one and whereas some others will cause white blisters on the plant's leaves on the right, as the one on the right. There are now wealth of commercially available fungicides in the market that help treat plant fungal diseases, but they share three common drawbacks. First of all, many of them contain copper and sulfur, which are toxic chemical elements that pose a threat to several microorganisms which are part of the natural ecosystem. Secondly, some of the organic fungicides contain chemicals that are highly soluble in the water, which make, so that they may run off into the local water system, contaminate the water, and thereby spreading the pollution. And finally, many of these fungicides, the environmental impacts associated with some of these fungicides are not well investigated, so there will always be a risk associated with the use of these fungicides. And inspired by these drawbacks, I began my research to explore the fungicidal capability of three options of alternative fungicides to figure out whether they are both effective as fungi and environment friendly. So the three alternative fungicide options that I chose, as you can see on the slide, are Tabasco hot sauce, Tide detergent, and Windex. You may wonder why I chose these compounds instead of others, other than the fact that they're all very common household products, and here are the reasons why. Tabasco hot sauce contains the chemical capsaicin, which is the chemical that gives hot chili the spicy taste. And there have been studies showing that capsaicin has some sort of antibacterial effect, which means that it kills bacteria. And I'm interested in whether the same result extends to fungi. Windex contains a prominent amount of ammonia, which is also toxic to yeast during certain life stages. And at the same time, ammonia is also an integral ingredient of many plant fertilizers, and as it furnishes nitrogen to a plant and poses no threat to the plant's health. And finally, Tide detergent contains borate ions and linear alcohol species, which is a group of chemical compounds that consist of a long hydrocarbon chain attached to a benzene ring. Both of these compounds are toxic to yeast, as they either inhibit key enzymes in yeast's metabolic pathways, or they destroy yeast's cell membrane. Borate ions is a mineral that can be utilized by plants. Linear alcohol species pose minimal threat to plant cells, and can be degraded under natural conditions, so both of them are very safe. At the beginning of my research, I set forth the hypothesis, which you can see in the white box on the slide. Since real parasitic plant fungi are really hard to maintain under laboratory conditions, I decided to use baker's yeast as my model organism, which is also a species of unicellular fungus. There are at least two variables that I have to consider in my design of experiment, which are the type of the treatment and the level of the treatment. And to take into account of both of these variables, I applied the three alternative fungicide options at different concentrations to different cultures of baker's yeast, and I measured the number of yeast colony that grow from each of these cultures. And here's a photo showing what yeast colonies look like. And this is what my experiments look like. So essentially, I applied each alternative fungicide at three different concentration levels at 10%, 1%, or 1% to saturate yeast cultures. And since this is a controlled experiment, the treatment group and the control group. So essentially, because I have one person, and the control group receives no treatment that does not have any effects on the growth of the yeast. So by having this set up, I can investigate the effect of fungicides on the growth of yeast alone while holding other conditions the same. And this is what I found in my experiment. So each of the data points from the graph represents the difference in a treatment control pair. So for example, in the graph on the left here, the leftmost data point, which you can see here, means that when applying the Tabasco hostas to yeast culture at 10% treatment level, the number of yeast colonies that I observed in the treatment group is about 40 less than the number of yeast colonies that I observed in the control group. And that, in other words, means that the treatment of Tabasco hostas at 10% reduced the number of yeast colony by about 40. And you may wonder if that's a significant reduction. Well, if you look at the 95% confidence interval that I draw here, you can see that it captures 0, which is the red line in the middle. And what this means is that statistically, 95% of the time, the difference between treatment and control will fall in a range defined by the upper whisker of the confidence interval and the lower whisker, which extends to the negative infinity. And this, statistically speaking, is not significantly less than 0. And since, as you can see, all the confidence intervals here shown on these two graphics capture 0. So we can conclude that both Tabasco hostas and Windex lack a significant adverse effect on yeast, at least under the conditions tested in my experiments. Well, the scenario is a little different for tide, as you can see here. For the 1% level, 10% level treatment and the 1% level treatment, the confidence interval do not capture 0. And that means that 95% of the time, the difference is going to fall below 0. That means that there's a significant reduction statistically. And the rate of reduction is about 94 to 95% in the case of the 10% treatment, and it's about 70% in the case of 1% treatment. And therefore, we can draw a conclusion that Tabasco hostas and Windex do not significantly adversely affect the yeast, whereas tide does, at least at 10% and 1% concentrations. And here are some pictures I take in my experiments. Just as every scientific study is limited in certain ways, there are also limitations to my studies. First of all, because due to logistic constraints, I can only make 4 replicates for each treatment scenario, which gives me limited data for statistical analysis. And because this is a small sample, it deprecates the accuracy of the statistical tests, which are the confidence intervals that you just saw on the last slides, and which may increase the chance of me making a mistake when judging the effect of alternative fungicides. And secondly, I used yeast matrices as my model organism in my study, which may or may not be representative of certain parasitic fungi species. And therefore, there's also limitations in the applicability of my research to true parasitic fungi under natural conditions. And therefore, I recommend field work involving applying tide to real parasitic fungi under natural conditions to be done before establishing it as a practically effective fungicide. And finally, here are some reflections about my research. It definitely helped that this project improved my lab skills because all the experiments I did in the project are about pipetting at very high accuracy levels. Also teaches me how different it was to work in a lab versus to read about science in a book. And finally, the project was a great exercise of applying scientific method and design of experiments to solve real-life problems, which I think is very interesting and very powerful. And that concludes my presentation.